Hello, good morning. Our today's topic is types of immunity. Immunity is a very important phenomena in living organism which provides protection against the pathogens. Immunity basically is of two types: innate immunity and adaptive immunity. Innate immunity is the one which we get from our parents, whereas adaptive immunity we acquire through our lifetime with the interaction of the pathogen. So innate immunity is provided by, for example, the epithelial barriers. The skin, which is jack of all trade, it will not allow the microbes to enter through it and cause infection. Then is the phagocytes. The blood cells have neutrophils and monocyte, which changes into the phagocyte. They engulf the foreign particle or the bacteria and nullify them. Next, another innate immunity example is the NK cell, natural killer cells, which are activated with the presence of bacteria and kill them. Next is the complement system. We have about 30 complement proteins in the blood plasma. These proteins, they causes lysis, they causes inflammation, they causes phagocytosis in the body system and protect the body from the various pathogens. Now coming to the adaptive immunity. For the adaptive immunity, B cells and T lymphocytes, they play a very important role. When any bacteria comes, it is received by the B cells. The B cell receptor, they will bind with the bacteria and get activated, and they lead to the production of large number of antibodies. These antibodies, they are Y-shaped proteins and they neutralize the bacteria or the pathogen and are responsible for eliminating it. The T lymphocytes, they cannot directly bind with the antigens. They need antigen presenting cell. So T cell receptor on the T lymphocytes bind with either MHC class 1 or MHC class 2 molecules containing the processed antigen, and with the help of either the cytokines or the perforins, they inactivate or kill the bacteria or the antigens. So initially the innate immune response is initiated. Later on the adaptive immune response is initiated inside the organism. So innate immune response is also known as natural or naive immunity. It refers to defense mechanisms that are present even before infections and have evolved to specifically recognize microbes and protect multicellular organisms against the infections. Innate immunity is the first line of defense because it is always ready to prevent and eradicate the infections. The major component of innate immunity are epithelial barriers that blocks the entry of environmental microbes, phagocytic cells, mainly the neutrophils and the macrophages, natural killer cells, and several plasma protein, including the proteins of the complement system. The phagocytes are recruited to the site of infection, resulting in inflammation, and here the cells ingest the microbes and are then activated to destroy the ingested pathogens. The complement proteins are some of the most important plasma protein of the innate immune system. Now coming to the adaptive immunity. Adaptive immunity, also known as acquired or specific immunity, consists of mechanisms that are stimulated by microbes and are capable of recognizing non-microbial substances also. Adaptive immunity develops later after exposure to microbes and is even more powerful in combating with the infection. By convention, the term immune response refers to the adaptive immunity. The adaptive immunity consists of lymphocytes and their products, including the antibodies. The receptors of lymphocytes are much more diverse than those of innate immune system, and lymphocytes are not inherently specific for microbes, and they are capable of recognizing a vast array of foreign substances. In the remainder of the introductory section, we focus on lymphocytes and the reactions of the adaptive immune responses. There are two main types of adaptive immunity. Number one, cell-mediated or cellular immunity, which is responsible for defense against intracellular microbes. Second, humoral immunity, which protects against extracellular microbes and their toxins. Cellular immunity is mediated by T lymphocytes, and humoral immunity is mediated by B lymphocytes and their secreted products. All these mechanisms of adaptive immunity are capable of causing injury to host and subsequent disease. Now coming to the difference between the humoral and cell-mediated immunity. In the case of humoral immunity, it is associated with B lymphocyte and is responsible for destroying the pathogen by producing antibodies against it. Whereas cell-mediated immunity is associated with the T lymphocytes and is responsible for destroying the pathogen or microorganisms which have invaded the cells. Humoral immunity is intimately associated with B lymphocytes, T lymphocytes, and macrophages, whereas cell-mediated immunity is associated with T lymphocytes, helper T cells, natural killer cells, and macrophages. Humoral immunity plays a major role in recognizing antigen or any foreign particle and in producing antibodies against it. Cell-mediated immunity is related to T lymphocytes, which work by identifying viruses and microorganisms and destroying them by the cell lysis or phagocytosis or pinocytosis. Humoral immunity secretes antibodies, whereas cell-mediated immunity is known for working against intracellular pathogen, and it secretes cytokines. Humoral immunity is rapid and quick in their response. Cell-mediated immunity shows delay through permanent action against any pathogen. Humoral immunity is involved in the early stage of graft rejection due to the formation of antibodies. Cell-mediated immunity is involved in the rejection of organ transplants. Now the humoral immunity, it mediates hypersensitivity type 1, 2 and 3, whereas cell-mediated immunity, it mediates hypersensitivity type 4. So this is the basic difference between the humoral and cell-mediated immunity. So in this diagram we can say that the humoral immunity, it works through the B lymphocytes. The antigen it activates the B lymphocytes. Different B lymphocytes, they are activated by different types of antigens. So they will produce the antibodies. These antibodies, through neutralization, lysis, and phagocytosis, are responsible for eliminating the pathogen. Now coming to the cell-mediated immune response, T lymphocyte is involved in the cell-mediated immune response. T lymphocytes cannot bind with the antigen directly. They need antigen-presenting cell. That is, the antigen, it enters into the macrophage or the dendritic cell. It is processed there into small peptides. These peptides, they will come out on the MHC molecule, and then the T cell receptor of T cell binds with it. So we have got two types of T lymphocytes, T helper cells and T killer cells. T helper cells, when bind with the antigen presenting cell, they produces cytokines. These cytokines will either activate the B cells for enhancing the production of antibody, or activate the T killer cell for enhancing the production of perforins for attacking on the infected cells. Now the MHC class 2 molecule containing the peptide binds with the CD4 receptor of T helper cells. T helper cells, when activated, produces the cytokines. These cytokines activate the B cells. The B cells, they once activated, they will lead to the production of clones of plasma cells and the memory cells. Memory cells, they are there for the future use, whereas the plasma cells, they will produce large number of antibody molecules, and these antibody molecules bind with the antigen and neutralize it. So this is regarding the cell-mediated immunity. I hope the concept is clear. Thank you.